This section explains why a virus cannot be defined by what it is made of and what single axis organizes the entire study of virology. A virus resists definition by substance, so Lostra defines it by constraint. 4 traits, jointly exclusive, name the set, an obligate intracellular parasite with an inert extracellular virion encoding at least one capsomer, reproducing by assembly rather than division, and capable of evolving. Nothing else on Earth holds all 4. But the chapter's real primitive is the organizing axis it hands the rest of the book. Every virus, whatever its genome, must reach the host ribosome, which reads only messenger RNA. The Baltimore classes are just the 7 distinct paths a genome can take to become messenger RNA. The taxonomy is functional rather than ancestral, genome to messenger. RNA distance predicts the entire replication strategy. This section explains the primitive the whole book runs on. A virus is a verb. It is defined by what it does to a cell. It enters, it commandeers the host's macromolecular machinery. It replicates, it kills. Chapter 1 hands you that primitive. Every virus, however diverse its shell or genome, faces one universal bottleneck. It must route its nucleic acid to messenger RNA because the host ribosome reads nothing else. Classify by that path, and the bewildering zoo collapses into the Baltimore classes. The spine of the book, chapters 5 through 10, is just each of those paths in turn. So the taxonomy is behavioral rather than morphological. You sort viruses by the problem they solve, getting their message onto a ribosome, and the diversity is the set of solutions. This part explains why a virus reproduces differently from a cell, and what that one difference generates. A cell reproduces by dividing, one continuously becomes 2. A virus reproduces by assembly, it disintegrates into free parts inside the host, floods the cell with copied genomes and proteins, then hundreds of progeny snack together at once. That one structural difference generates the whole chapter. Between entry and burst sits the eclipse. No intact virion exists anywhere. The particle has literally ceased to be. That is why the one-step growth curve stays flat, then goes vertical. Decompose that arc, and 6 universal stages fall out, attach, penetrate and uncoat, early proteins, late proteins and genomes, assemble, release. Same tune in every virus, individual flair at each step. Chapters 3 through 11 take 1 stage each. This section explains how a virus reproduces and why that mode differs from the way a cell copies itself. A virus reproduces by disassembly and then mass assembly, never by the growth and division every cell uses. The TEL is the eclipse period, that interval after infection when not one infectious particle exists anywhere. A dividing cell splits into 2. It never vanishes and reappears. During eclipse, the virus persists only as decomposed instructions running on hijacked host machinery. This is why Ellis and Delbruck's one-step growth curve stays flat and then erupts. The chapter's whole claim is that this cycle factors into 6 universal stages: attachment, penetration, gene expression, genome replication, assembly, release. It is one pipeline that every virus executes with its own flair. One boundary structures the rest of the book. Lytic reproduction kills fast. Latency defers the same program indefinitely. This section explains how a virus gets inside a cell across 3 steps: attachment, penetration, and uncoding. Entry is a lock and key filter, and the key decides everything downstream. Before any viral gene is read, a virion must find a cell whose surface receptor its capsomers or spike proteins physically complement. That single complementarity, not the genome, sets which species, which tissue, which cell is susceptible. Attachment is therefore recognition, and recognition is therefore recognition, and recognition is destiny. Penetration then converts binding into breach. A fusion protein in enveloped viruses or a capsid pore in naked ones sits preloaded with strain energy and fires a large conformational change when the cell's own cue trips it, either receptor contact or endosomal acidification. That firing dumps the genome inside, expression ready. The shape of the whole thing is a specificity gate, then a triggered spring. This section explains what sets a bacteriophage's whole replication strategy, and the answer is a single variable, the chemistry of the virus's genome. That one property generates everything downstream, which polymerase runs, how expression is timed, and how a tiny genome manages to encode enough protein. The reason it controls so much is the host. The host cell reads only one pipeline fluently. Double-stranded DNA becomes messenger RNA becomes protein. Whatever that pipeline cannot do for a given genome, the virus must supply for itself. So the chapter holds the organism fixed, always E. coli, and runs it against 3 genome types: double-stranded DNA in phages T7 and lambda, single-stranded DNA in Phi X174 and M13, and plus-strand RNA in MS2 and Q beta. Each phage's entire strategy falls out of one question: how far is this genome from the hosts? Entire strategy falls out of one question: how far is this genome from the host's native? This is the template for chapters 5 through 10. The Baltimore logic, that genome type predicts mechanism, is being installed here, on the simplest hosts. This section installs the primitive, that a virus is nothing more than its genome. A virus's genome is its entire program. Everything it must borrow, encode, or subvert follows from the chemistry of that genome alone. That is the primitive this chapter installs. Name the genome type: double-stranded DNA, single-stranded DNA, or positive sense RNA, and you can predict the expression and replication strategy before you ever meet the expression and replication strategy before you ever meet the organism. Lostrow proves it on the simplest interpreter, the bacterial cell, using 6 phages as anchors: T7 and its uniquely viral RNA telomerase, lambda and its lysogenic switch, Phi X174 and its overlapping reading frames, M13 and its replicative form, staging, MS2 and its structure-gated translation, and Q beta and its structure-gated translation, and Q beta and its uniquely viral RNA polymerase. Each one lights up a single facet of the same law. This is the first of the Baltimore organized chapters, 4 through 10. The harder eukaryotic host is where the same law reruns. This section explains why the positive strand RNA viruses behave the way they do, derived from 2 host constraints. The positive strand genome is already messenger RNA, coding sense. So the first act after uncoding is its own translation. No polymerase needs to ride inside the virion, unlike the negative strand and DNA classes to come. That single fact makes the whole chapter derivable from 2 host constraints the virus must defeat. First, a eukaryotic ribosome reads 1 protein per message, gated by a cap and a poly, a tail with no internal shine dalgarnocyte. So a monocistronic genome that must yield many proteins forces every escape trick you will meet: polyproteins cleaved by viral proteases, IRES entry, frame shifts, and leaky scanning. Second, No host enzyme copies RNA from RNA, so the virus must translate its own RNA-dependent RNA polymerase, the RDRP, before it can replicate. Given those two constraints, predict the family. This section explains why the negative strand RNA viruses form a single class and how one enzyme generates their whole way of life. A negative sense genome is the photographic negative of its own messages. It is complementary to every messenger RNA, so it is untranslatable on arrival. The host ribosome finds nothing to read. This gives the constraint that generates the whole class. The virion must carry its own polymerase inside, packaged together with the genome, because the first enzyme cannot be built from a template that cannot be read. That RNA-dependent RNA polymerase then reads one template two ways. Obeying the start-stop punctuation at the gene junctions, it emits capped polyadenylated messenger RNAs in a quantity gradient set by gene order. This is its role as transcriptase. Ignoring that same punctuation, it copies the genome whole into an antigenome. This is its role as replicase. Rabies, measles, Ebola, and influenza, with its cap snatching, are each a dialect of that single toggle. This section explains why a negative sense virus must carry its own copying enzyme and how one enzyme does two opposite jobs. A negative sense genome is instructions written backwards, inert, untranslatable as it stands. So every classified virion must smuggle in its own polymerase. Without it, the genome does nothing. The chapter's real compression sits deeper. That one packaged enzyme reading one template performs two opposite jobs. It either excises short cap messenger RNAs or it copies the genome whole, and a single scalar picks which. Free nucleocapsid protein is the dial. When it is scarce, the polymerase stutters at gene junctions and releases discrete messenger RNAs. When it is abundant, it coats the nascent strand, and the polymerase reads straight through into a full-length antigenome. Transcription and replication are one machine, not two, switched via concentration. This section explains why double-stranded RNA viruses are built around a single defensive constraint. A genome that is itself the enemy's alarm can never be set free. Host cells scan the cytoplasm for double-stranded RNA as the unmistakable signature of viral infection, so a virus made of double-stranded RNA meets a lethal constraint, expose the genome, and die. The whole chapter is the single consequence of that constraint. Never fully uncoat. Rotavirus keeps its genome sealed inside a partially intact capsid, the double-layered particle, which doubles as the reaction vessel. The very structural proteins of the shell are the transcription and replication enzymes. Only capped messenger. RNA is extruded through the vertices. The double-stranded RNA never leaves. Architecture dictated by what the host is listening for. This section gives the one pattern the chapter turns on. A double-stranded DNA virus carries a genome chemically identical to its host's, and that sameness is both gift and trap. The gift, its genes plug straight into the host's own nuclear transcription machinery and express for free, with no polymerase of its own needed. The trap, the cells it infects are usually terminally differentiated, so their DNA replication enzymes are switched off. To copy its genome, the virus must reawaken them, forcing the cell into S phase. Expression is borrowed. Replication is stolen by driving the cell cycle. The pox viruses are the exception that proves it, carrying their own enzymes to replicate in the cytoplasm. This section explains the defining constraint of double-stranded DNA viruses that infect animal cells. A double-stranded DNA virus carries a genome chemically identical to its host's, so it hands transcription straight to the host's own RNA polymerase and pays almost nothing to make its proteins. The catch is replication. The cells these viruses infect are usually terminally differentiated, and such cells keep no DNA replication machinery on hand. So the defining move of the entire class is coercion. It forces the resting host cell into S phase, and the replication proteins the cell builds for itself become the viruses. From that one constraint, you can predict the exception. A virus, too large to obey the host's rules, must carry its own enzymes and replicate in the cytoplasm. That is the pox virus. This section gives you the single idea the chapter turns on. A virus can be the shortest program that, dropped into a running cell, rebuilds itself. Circoviruses and parvoviruses carry single-stranded DNA genomes smaller than one human gene, yet they run a complete infection with fewer than 6 proteins. The move is offloading. They encode only what the host cannot supply, recognizing an origin, nicking a strand, packaging the copy. Everything else, they let host polymerase and host transcription do. Everything the cell can already compute is left uncoded. What remains is the irreducible difference between virus and host, a genome compressed to its informational floor. This section names what Chapter 9 is for, the extreme case of genomic minimalism. Circoviruses and parvoviruses run a complete infection from a genome smaller than a single human gene, encoding fewer than 6 proteins. The organizing trick is delegation. A single-stranded DNA genome cannot be read by host RNA polymerase, which demands a double strand, so host enzymes first copy it into double-stranded form. And from there, the host machinery does the transcription. The virus ships a program without its interpreter and borrows the cells. Every sequence then pulls double duty, overlapping reading frames, alternative splicing, and terminal secondary structure that serves at once as origin and as primer for copying the genome. This section explains how a single enzyme binds 2 viruses that share nothing else. One enzyme rewrites the arrow. Reverse transcriptase reads RNA back into DNA, and the whole chapter is the claim that this one inversion is what binds 2 otherwise unrelated viruses. Not shared ancestry, not shared ancestry, not shared ancestry, not shared structure. The 2 are HIV-1, a retrovirus arriving as GOPT DNA, and HBV, a hepadnavirus arriving as GAPT DNA. Classify by the enzyme that reverses information flow, and the rest follows. There is no proofreading, so the mutation rate runs roughly 10,000 times the host's, and the population swarms into diversity. The DNA intermediate must integrate or persist. The drugs hit this polymerase while sparing the host's own reverse transcriptase. The understated surprise is downstream. The same inversion is written into your genome as endogenous retroviruses and reused to repair the ends of your chromosomes. This section explains why 2 viruses with nothing in common on the surface belong to a single class. Consider HIV, A+ strand RNA retrovirus, and hepatitis B, a partly double-stranded DNA hepadnavirus. On the surface, they share almost nothing. They collapse into one class the instant you ask which way information flows. Both run RNA into DNA through a viral reverse transcriptase, inverting the arrow that the rest of biology treats as fixed. That one reversed arrow is the classifier, not the surface form of the genome. The divergent details, a tRNA primer versus a protein primer, integration or no integration, all hang off it. This is the chapter's job in the book's Baltimore class spine. It is the reverse transcribing class. The generative move is to sort by the direction of the copying map, not by what the genome looks like. That move is the whole chapter compressed into one primitive. This section closes the replication cycle by explaining how the finished virus particle comes together. A virion condenses. Nothing assembles it from outside. Embryons reassemble unbidden, spontaneously. The host cell, stuffed with loose parts, capsomers, genomes, spikes, and the infectious arrangement, is simply their free energy minimum. Nations in effintensis, nations re minima. Mix purified TMV. That is the whole move. The assembly instructions live in the parts, not in a builder. This is exactly what severs viruses from cells. Omnis cellula, a cell continues a template by dividing. A virus has no parent to divide. Two codas follow. Release comes by lysis or by budding, and in some viruses, an irreversible maturation cleavage arms the finished particle. This section explains what a virus is doing during lytic growth and why the chapter's 12 interactions all reduce to a single move. A virus is a resource parasite racing its host's death. Every interaction, the chapter catalogs, degrading the chromosome, cleaving a translation initiation factor, blocking or triggering apoptosis, hijacking membranes, is one and the same move. Redirect a host program from host benefit to virus. The chapter's compression is that where you strike is dictated by host architecture, not by virus taste. Bacteria couple transcription and translation in one compartment with messenger RNA that lasts about 5 minutes, so phages kill transcription. They degrade the chromosome, and T4 spares its own DNA through hydroxymethylcytosine. Animal cells split the nucleus from the cytoplasm with messenger RNA that lasts hours, so their viruses strike translation directly. From this, you can predict any new virus's tactic from its host's floor plan. This section explains the single structural move that every trick in the chapter is a rotation of. A virus carries no factory, only a genome and a coat. So each trick is one move, seen from different angles, hijack a host subsystem. The virus cannot build itself. Translation, apoptosis, protein degradation, autophagy, the stress response. Each one is captured, never attacked for its own sake. The governing constraint is a race against a trade-off. Shut host synthesis down to free ribosomes and starve antiviral proteins, yet keep the cell alive long enough to assemble thousands of offspring. Whether a virus strikes at transcription or at translation itself is derivable rather than arbitrary. It falls out of two host parameters, the half-life of the messenger RNA, and whether transcription and translation share a compartment. This section explains why a virus need not kill the cell it enters. That is the whole move of this chapter. Set that against the book's default picture, the obligate lytic machine which runs 6 stages and then bursts its host. Persistence is a virus decoupling its own continuation from the cell's death. It holds its genome inside without finishing the cycle. The generative primitive is a bistable switch, replicate now versus right along, whose set point means host condition. Bacteriophage lambda is the canonical instance. Stress stabilizes the protein. C2 throws the switch toward lysogeny, and comfort throws it toward lysis. Latency is the switch held reversibly open. Chronic infection is the same switch leaking slowly for a lifetime. This section explains persistence, what a viral program becomes once it stops killing its host right away. Every prior chapter watched a virus do one thing: enter, run all 6 stages, kill the cell now. Persistence is what that program becomes. Once replication stops being immediate, the genome lingers, decoupled from virion production. There are 2 ways to linger. One is to keep lysing slowly, faster than the immune system can clear you, as in HIV and hepatitis. The other is to leave the lytic cycle entirely and idle as a few nucleic acids, reversibly as in lysogeny and latency. The single move that carries the whole chapter is this. Commitment to lysis becomes a switch keyed to host state, held toward quiescence while the host thrives, thrown back toward lysis under stress. Cancer is the collateral of long residence. The ordinary lytic virus is just this switch welded open.